So TME profiling is definitely something that has been very successful. One example is Lymphomabs, as published by Michael Green's group last year. So we do think that similar strategies to Lymphomabs, for example, could be used to predict response to bispecific antibodies, including in the first-line setting. This research, it provides deep mechanistic insights. However, we rely too heavily on multiplex immunohistochemistry and on the localization of these different immune cells, which makes it harder for direct clinical application. But this is the goal that we can achieve, to use the tumor microenvironment to predict which patients are going to respond or not to Glofitamab therapy. Now, if we can use the tumor microenvironment profiling to predict whether a patient will respond to stratify a patient for CAR T cell therapy or bispecific antibodies, that's the ultimate goal. Of course, for that, we will require a large number of patient samples and longitudinal patient samples, and then assess patients who don't respond to CAR T cells and then respond to bispecific antibodies, or vice versa, and the reasons why they can respond to one and not to another one. But in my opinion, the most important thing will be to see where the T cell engagers will be used, will end up being used in the therapy more often. Because if they make it to the first line setting, the key question will be to understand why patients who fail bispecific antibodies also fail CAR T cells. So it will be to identify these patients and then work out what would be the best therapy for these patients, or what would be the best combination therapy for these patients.